Okay, well, hello everyone from Perth. My name is Jaume and I'm a PhD student at the University of Western Australia. And today I'd like to share with you some of the main findings from my PhD thesis, which basically arises from the need to find quicker and more efficient ways of measuring plant condition in a restored mine site. Now, there are very well developed and known methods that are very precise to measure plant condition. However, some of these methods are sometimes difficult to perform, and they require destructive sampling and lab work, which makes it difficult to use them in very large areas where we're looking at monitoring a very high number of plants. So the idea is to see if remote sensing methods can assist in measuring plant condition in a more efficient way. Now, remote sensing can be done at very different scales, depending on the distance between the sensor and the object of interest. It can be done from space, or it can be done from the air, and it can also be done from the ground. And in this project, I focused in using ground level methods, basically because they provide a level of accuracy that is appropriate to establish those relationships between physiological traits and remote sensing. And the idea is to see which of these methods have potential to be upscaled to more remote platforms. So a lot of these methods have already been developed and implemented and even automated for agricultural environments. However, agricultural environments are very different to what we're talking about today. They're very uniform and they're normally composed of only one plant species. Whereas we're looking at something that looks like this, which is an open environment with a very diverse range of plant species, forms, functional types. So it's going to be a bit more difficult. So in my project, I focused in two specific remote sensing methods. The first one is hyperspectral reflectance. So it's been well established that objects reflect light differently depending on their chemical and physical characteristics. And this is the case as well for plants, both at the leaf level and at the whole plant level. So we basically measured plant reflectance, and we did that using a field spectrometer, which is this device, which is a very, very precise piece of equipment that gives us point information. So it doesn't generate an image, but it gives us point information. But this point information is very precise, and we get information on all the bands, 1 nanometer bands in the optical electromagnetic spectrum. And the second method we used was temperature measure. Now, the relationship between plant temperature and physiology is that when plants are photosynthetically active, they open their stomates and they transpire water. And when that happens, this water evaporates on the leaf surface and the leaf cools down. So a plant that is healthy will have a lower temperature than a plant that is stressed and has shut down its stomates and is seeing that temperature is increasing. And the way we measure that is through a thermal camera, a handheld thermal camera. And the advantage of these in front of other temperature measuring devices is that thermal camera provides the information of temperature on the whole leaf and also on a group of leaves if you're interested on that in a very quick way. So in my experiment, I grew plants in a glasshouse from 10 different species. Now, these species were selected because they had different strategies to adapt to drought. So what we did is we sort of artificially droughted them and took measures along the droughting process. And these plants, as you can see here, have very different leaf morphologies, which was also part of the study's objectives, was to see whether these methods could work with such a variety of leaf and plant morphology. So in this first experiment, we basically tried to see if hyperspectral reflectance can estimate water status specifically. And we used both the traditional methods there represented on the right and the hyperspectral reflectance values measured at the leaf level with the leaf clip and also at the whole plant level using a spectrally flat background. And specifically what we did is we measured 4 different physiological variables there on the right. and tried to relate them to 4 different methods to analyze hyperspectral data. And what we found is that, first of all, there were no differences in model performance between broad-leaf species and narrow-leaf species, both at the leaf and the plant level. Now, that is quite relevant when we're looking at characterizing plant communities as diverse as the ones that we have here in Australia. Secondly, shortwave infrared regions in the spectrum were the most informative in terms of providing information on water status. And another aspect we found is that models were able to separate healthy plants from stressed plants, but they were not so precise at detecting those initial stages of stress in which symptoms are still low to moderate. So that could be a limitation of this method. In our second experiment, we used those same droughted plants to try to estimate stomatal conductance, so how much water are plants transpiring, using thermal imagery that you can see there on the left. And on the right, you can see a Lycor, which is the device that is normally used to, or is one of the devices that can be used to measure plant transpiration and photosynthetic activity. And the way we tried to relate these 2 sets of data was basically, we used 2 different approaches using the temperature data. One was to try and model the leaf energy balance. Through this approach, you can basically estimate stomatal conductance directly. And the other approach is to use wet and dry reference leaves in the same image as your sample image to generate an index that is proportional to stomatal conductance. And what we found is that we were able to actually model, using that leaf energy balance, we're able to model stomatal conductance quite accurately for 6 out of the 10 studied species. Now, our estimates were mostly overestimates, and that was caused by inaccuracies in determining leaf boundary layer characteristics. And leaf boundary layer is a small, thin layer of air that is surrounding the leaf, and that's the layer of air with which the leaf is actually interchanging energy. But it's very hard to model how thick it is, and that is definitely a limitation of this method. However, we also found that the proportional index of stomatal conductance was actually pretty good at relating to changes in actual conductance. So it can be used in comparative studies in the field where we have information of plants that are very stressed and plants that are very healthy, and we're able to put our sample plant in context of those 2 sort of upper and lower limits. In our experiment, we said, okay, well, we're gonna try and see whether these methods that seem to work reasonably well in the glasshouse, how do they work in real life conditions? So we went out to a restored mine site here, 40 kilometers north of Perth, and we tracked seasonal change in 5 different species of plants using the traditional ecophysiological methods that are able to basically characterize that seasonal change very precisely. And at the same time, we took the remotely sensed measures and we tested whether those variables, those proxy-derived variables, are able to inform of this seasonal change reasonably well in comparison to the traditional ecophysiological methods. So there, basically, you can see the variables we measured on the left through the traditional ecophysiological methods, which were the same we measured in the glasshouse. And we added a couple more that we also know that are also known to be able to be derived through remote sensing, such as fluorescence and several pigment contents there. And yeah, sorry, these are the 5 species we studied in the field, which are basically very different functionally. Some of them deal with drought by avoiding it, by dipping deep root systems, whereas others just tolerate it. So we want to see how these methods work in very different plants, and they all have very different leaf shapes as well. The first thing we did is we looked at univariate relationships. So how does a physiological variable there in the y-axis of those plots relate with a spectrally derived variable in the x-axis. Here I'm showing information for 2 species, which are the 2 rows, and each one of the columns is a different physiological variable that we're trying to approximate using remote sensing. So as you can see, some of these methods work well for these both species, and some are a bit more variable. And I want to reiterate the fact that this is data at the leaf scale only. I will be telling you a bit more about what happened at the plant scale later. So we have these reasonably good univariate relationships, although very variable. So our next question is, can we actually combine them and track seasonal change? So the first thing we did, and that's all done through multivariate statistics, was we used the physiological data set to sort and group the samples by seasons. They're shown in different colors. And when we use the physiological data, we can see that seasons, samples from the same season sort of grouped together. There is some overlap, but we are able to separate them reasonably well. And we did the same thing using the remotely sensed data, or what we're calling these proxy variables. And we can see how the separation is also reasonably good, although there is some overlap. You can see that early and late summer are mostly clumped together, whereas spring and late winter and autumn also appear together. There's another step on this analysis that we can do, which is to look at the season centroids, which is basically sort of like a summary average point for each season, and where that sits within this multidimensional space. So the closer 2 points are on this space, the more similar they are, and the farther away, the more different. And so you can see how the arrows there are indicating the change from one season to the next one, and the results for both data sets are actually quite similar. And the next thing we can do is we can actually measure the distance between these points and then compare both approaches, which is what this plot shows here. On the x-axis, you have the physiological distance between seasons, and on the y-axis, you have the remotely sensed distance between seasons. And you can see for this particular species, the relationship is actually very good, which means that the remotely sensed data is actually to track changes throughout the season, very similarly to how the physiological data is doing. So the main results of this third experiment is, first of all, as I mentioned earlier, I wanted to talk to you about the plant scale. So things didn't really work at the plant scale, basically because we're holding this fiber optic on top of a plant that has a very variable background. And in many cases, these plants have very sparse canopies, which means that we don't have too much plant tissue in our signal, and the soil is very variable. So it's definitely, it's adding a lot of noise. So those univariate relationships didn't really work, which prompts more research is needed in this scale. However, at the leaf scale, we did find some significant relationships when we look at univariate variable comparisons. And when we bring them together, we see that these remotely sensed proxy indicators are able to track seasonal changes in 4 out of the 5 studied species in the field. So as conclusions of my thesis so far, we can say that it is possible to detect drought stress in a diversity of plant species that grow in a restored mine site of WA using remote sensing. Now in the field, this is only possible at the leaf scale, at least for the species that we studied and for the age of the plants that we studied that were very young, 2 to 4 years old. So we would have to see how that would work at the scale if we're looking at older plants that have much more plant tissue to be detected by the spectrometer. Individually, these studied methods can accurately detect differences between healthy and plants in which stress symptoms have been fully developed. However, they're not so accurate when it comes to detecting those initial stages of stress. But when we bring them together in a multivariate analysis approach, these methods that we study actually can accurately track seasonal change for different functional types. And this is where I'm at the moment with this. I'd like to thank the Center for Mindside Restoration for supporting me in this research, and Hansen Construction Materials, which allowed me to do fieldwork at the site, as well as my supervisors for their ongoing support. And thank you all very much for your attention.
Yeah, so just a quick question about the imagery that you're conducting. Do you need to have the plant identified before you're doing those, taking those images? Like, does that, I was just wondering, is there a potential for that technology to be adapted to drone usage so that you could just do an aerial flight over a site and then analyze which plants are undergoing what sort of level of stress from drought conditions?
Yeah, so I need to clarify that I was only taking images for the thermal side of things. So I was taking images of my leaf of interest, and with the spectral side of things, I was using a spectrometer, which, like I said, it doesn't provide an image, it provides point data. Now, in terms of upscaling these to, like you suggest, using drones, so there's different ways of doing it. So, for example, one of the things we identified was that we're able to select a specific wavelengths that, when combined in the form of a spectral index, they can be very informative of water status for certain plants. So once you have those wavelengths identified, for instance, an approach would be to select filters for those wavelengths and mount them on a multispectral camera, and then mount them on a drone that would basically give you an image of that specific spectral index that you've identified as being related to water status in this case. Now, you probably would need high enough resolution for you to be able to separate individual plants from each other. Whether you do that through like object-based analysis, or you first tag your plants in the field, that's something that is sort of beyond of the scope of my research. But basically what we're trying to do here is we're just trying to see what has potential to be upscaled to more remote platforms. That's sort of what we're trying to do. I guess this doesn't really answer your question, but it clarifies that we were not really dealing with aerial imagery.
It's still